Well, welcome back. So here we are in the final chapter, chapter 17. We've looked at a lot of different aspects of the microorganism, including what the structure is, what the functions are of the different structures. We've looked at their metabolism and their genetics. We've looked at how to prevent infections from spreading with microbial control. We've looked at how to treat infections. We've looked at how the microbes attack the host and what do they do, how do they cause damage, how do they establish infection and cause damage, and then how do they leave, how do we transmit infections. We've also looked at how the immune system fights back. And so now we're going to be looking at another aspect of the clinical aspect of microbiology, and that is diagnosing infections. And so without further ado, here is Chapter 17, Diagnosing Infections. Chapter 17, Diagnosing Infections. So in this section, we're going to discuss the three major categories of microbial identification techniques. And by the time you're done, you should be able to provide a one-sentence description for each of these three categories. And for the test, you should be able to identify different tests that we have done or that we talk about and tell which type it is, which category it fits into. So for identifying the genus and the species, we look at phenotypic, immunologic, and genotypic. Those are the three categories. And so a phenotypic is looking at the phenotype, the expression of the genotype, basically. And so these are the microscopic and macroscopic morphology, physiology, and biochemical tests. So these are most of the things that we have done in the lab. Now we have done some immunologic tests in the lab as well, and that includes identifying a microbe using antibodies from the patient or testing patient's antibodies against prepackaged antigens. And so we're going to focus mostly on that in this particular section because a lot of the testing in the laboratory, when you send them out to the hospital laboratory, that's most of what you're going to be doing in today's workforce. So the genotypic methods are identifying an analysis of the microbe's DNA or RNA. And so you may be using immunologic methods, but if they're just specifically looking at something like this, which is a fingerprint, basically DNA fingerprinting of the microbe, then that is genotypic. So if you use antibodies and do a stain using antibodies, that's immunologic. But when you just look at the banding patterns here and you're looking at the sequence of the DNA, then that is genotypic. It's kind of a blurred line right now because a lot of these tests are using both. There are many ways and places in the body and outside of the body that we can collect specimens from. You can see the nasopharynx, which really is not just the nares. It goes all the way back through the nasal cavity, almost right there to the pharynx. And so it's pretty deeply invasive, let's just say. And if you've ever had one of those tests, you know exactly what I'm talking about. And then they can also swab the throat. They can also take sputum samples and saliva samples, blood samples, of course, and urine samples are pretty common. And then sometimes they'll take samples from catheter. They'll take skin and swab it and grow up the cultures. They'll take spinal tap, and that means they're sampling the cerebral spinal fluid. They'll go in between the vertebrae and pull out some fluid from the cerebral spinal fluid. And it is pretty invasive. It's not something you can do this at home for sure. You cannot do this at home. You should not do this at home. So feces is obvious. They can take that and grow up samples from that. Vaginal swab or stick, same thing. You can grow up samples from that. And then they'll take scalpels to scrape off skin from the foot or from other parts of the body. But those are some different ways that we can collect specimens. And then sometimes we need to transport the specimen, and so we have sterile transport swab within a container here. Sometimes it's called a culturette. And so it has a tamper-resistant seal, a plastic case around the swab that's a long swab with a rayon tip, and then this transport media. You pull the swab out, collect your sample, put the swab back in here, label it, identify what it is, and then you squeeze this tube and it releases medium that will keep the sample in kind of a stasis kind of thing, and it'll last about 72 hours. So that is our method of kind of transporting it. So like, let's say you have to go out into the field and you're collecting all these samples, and then you have 72 hours to get them into the lab and get them refrigerated, and then you can use them as you are ready for them. But this is usually used when you have to go to a particular site and sample many people, and you don't have time to get it right back to the lab and test it that day. So a couple of different, just an overview of laboratory techniques. We have the first step is to analyze the patient for signs of microbial infection. So look for heat, pain, redness, swelling, loss of function, that kind of thing. So fever, wound exudate, mucus production, abnormal lesions, those kind of things. And then you're going to collect specimens and you're going to analyze them. So direct tests are like microscopic, immunologic, genetic tests. And then other times we have to cultivate and isolate and identify pathogens. And the time range for the requirement for these analyses ranges from a few minutes to several weeks. So if it's a rapid test, it could take a few minutes. But if it's something that has to be grown up in culture and then there has to be other things done to it, it may take several weeks. So here's a clinical form used to report data from a patient specimen. So you can see some of the things that they have on a standard form, like where the source of the specimen, the test that they're requesting, and then the culture results. And then on some of these sensitivity tests, they'll do the regular kind of usual suspects and look to see the zone of inhibition. So notice that there's an A, B, and C, so we have like a we can see more than one test to be sure that we have an average that is correct. So in the next section, we're kind of skipping the phenotypic methods because we've already done that. And genotypic methods we'll talk about a little bit, but we're going to talk about the immunologic methods and mainly the serology. So what is serological tests? What's the term serology mean? And what's the principle behind the serological testing? And then we want to identify two immunological diagnostic tests that rely on a secondary antibody and how they work. So a serology is basically the study of the serum. So it's in vitro diagnostic testing of the serum. So you take people's blood, you separate it from the red blood cells, and what's left, the serum, should have antibodies in it. And so it's based on the concept that the antibodies have a very extreme specificity to the antigen, and then we have to visualize it. So visualizing this is what we call contact signal. And so a contact signal, you have some sort of a color or maybe a precipitate, maybe some bioluminescence or fluorescence, but something is going to help you to not identify that there is an antigen-antibody testing occurring, because of course the antigens and antibodies are very small, microscopic. It's not this isn't something you can see. So in serology, you take an unknown antibody and it can be detected using a known antigen, or vice versa. You can look at an unknown antigen and detect it with a known antibody. And so this is pretty much the key thing that I want you to know from this part or this section. So if you have an unknown antibody, then you're going to have a known antigen, and that is an indirect test. It's indirect because you're looking for antibodies in serum, and that is indirectly It's indirect evidence of exposure. So you may have like hundreds of antibodies floating around different that would detect different things. So it's indirect evidence of exposure. You could have a lot of different antibodies present, but if the one that you're looking for binds to a known antigen, then this identifies the fact that you have been exposed to it and you've mounted an immune response to it. So if you have antibodies present, you've gone past the incubation period, you have been into the prodromal, the acute stages, and you may be in that stage right now. Now, of course, there's a possibility that you have been exposed to it way in the past, but it would not be a really strong signal. And there are ways to know if it is something that is current or it is like from the very maybe recent past. It depends on the type of antibody. And as we learned in Unit 5, with the immune response, IgMs are produced first in the primary response, and then IgG is produced in the secondary response. It's produced first in the secondary response, and it is the antibody that's going to circulate around most of the time. And so with COVID, they have a test that can distinguish between IgG and IgM. But if you have an unknown antigen, then you have to have a known antibody, and that's the direct test, because what is unknown, you're actually detecting the antigen in your sample. You're not looking for evidence of it, you're actually detecting the antigen. So modern serological methods can use sera, so the blood, urine, cerebrospinal fluid, whole tissues, and saliva. So the basic idea is, again, do you have antibodies or antigens that you're trying to detect? So if you have a patient's serum, you're looking for antibodies in the patient's serum. If you have antigens, then you're like a culture, then you are going to detect it with known antibodies. So, but you have to have something that's known, something that's unknown. And so again, in this case, we are detecting, this is an indirect test. And where you are looking, so you are looking for antibodies. These are unknown antibodies and known antigen. And here, this is a direct test because you are taking unknown antigen and challenging it with different types of antibodies. So let's say you think you have a streptococcal infection, you could test it against group A strep, group B, group C, group D antigens. That would be, well, antibodies against those antigens. And so that would be a direct test because you've got a sample of the antigen and you just don't know what type it is. And you can have different antiserum. It's sort of like blood typing. You can test with different antisera that are antibodies against like type A, type B, type C, type AB, that kind of thing. Well, it's be basically type A or type B or the Rh factor. So visualizing, and again, this is something I mentioned before, you have to be able to see it with the naked eye, or it has to be evident in a light microscope. And so with whole cells, or whole human or microbial cells, they can form clumps, and usually those are visible. And so clumps or aggregates with antibodies, so whole human or microbial cells, but whole cells clumping with antibodies, and that is called agglutination, but that is something we can see. But with smaller antigen antibody complexes, you really may not be able to see it, so you have dyes or fluorescent reagents to be able to visualize it. So these are your contact signals. So we'll look at, first of all, the agglutination versus precipitation, because those sound like they are the same thing because they're clumping together, but they're slightly different, and they vary based on the size and the solubility and the location of the antigen. So with agglutination, so agglutination versus precipitation, you have these three differences. So size-wise, the agglutination is going to be larger. Precipitation is going to be smaller. And agglutination is going to be insoluble, where precipitation is going to be soluble antigen. And the location, the agglutination is going to be on whole cells, whether that's a microbial cell or a human cell. It's on whole cells, And precipitation is free-floating. In solution. And not attached to a cell. So I guess maybe just saying not attached to a cell would be probably more proper to say that. So that's one of the ways that we differentiate. And so we're going to show you a little animation on this now.